Great, so I'll jump over the introduction slide. So yeah, I'm going to be talking about our method called VESPA ASL. But before I get into that, I just wanted to give a little background about some relevant arterial spin labeling methods. So sorry if you're already very familiar with this. So specifically, I want to talk about a spatially selective ASL method called pseudo-continuous ASL, or PCASL, and also velocity selective ASL. So in PCASL, we define a labeling plane below our imaging region, typically in the neck, and we label blood as it flows through this plane. Because we label blood outside our imaging region and away from the tissue, there's a delay before the labeled blood reaches the tissue. And this delay is often called the arterial transit time. So to reduce sensitivity to this transit time, we use a post-labeling delay to let all of the labeled blood reach the tissue before we acquire an image. However, in some diseases, this transit time can be much longer than expected, or just much longer than the half-life of our label. And this can make getting quantitative or even qualitative information about cerebral blood flow very difficult or even impossible. So an alternative to spatially selective ASL techniques is velocity selective ASL. So in this case, we don't just label blood outside the brain or outside our imaging region, but also within our imaging region. And this means there's almost no delay before the label reaches the tissue. So we're not sensitive to delayed arrival like with spatially selective ASL techniques. So here's a demonstration of why that's important. So here's a Moya patient with a chronically narrowed right ICA, which you can see on the MR angiography image. The post-ASL image, which is also a spatially selective technique and suffers from very similar problems with transit times as PCASL, shows an area of apparent hypoperfusion on the right side of the brain. In contrast, the velocity selective ASL image shows no such area of hypoperfusion. And this is also corroborated by the DSE image on the right. So with the velocity selective ASL, we get a better understanding of the true tissue perfusion. But if we only acquire this image, we miss the important information contained in the PASL image, which is about the delayed arrival, and which we can infer from these hyperintensities, also called arterial transit artifacts, and argued to be very useful, particularly by people like Greg Zaharchuk. However, if we just acquire this PASL image, which does have this transit time information, we can guess that there's delayed arrival, but we don't know the true underlying perfusion of the affected tissue. So recently, Divya Bolar and his co-authors suggested collecting both sets of data so that you can more fully evaluate the relevant hemodynamics in cases like this, where there's very delayed arrival times. Unfortunately, this would double the required scan time for ASL, which can be tricky to fit into a busy clinical schedule. So then that brings us on to what VESPA ASL is. So in VESPA ASL, we combine the velocity selective and the PCASL labeling into the same scan, but we label different pools of water with each labeling technique. So the blood above the PCASL labeling plane, is labeled with velocity selective ASL to remove any transit delay issues. So we already have labeled blood filling the vasculature. And immediately after this, we start labeling with PCASL. After we've labeled with PCASL for some time, we use a vascular crushing module to remove any arterial signal. And now we have both spatially selective and velocity selective contrasts in our tissue in one scan. But we need to be able to separate these two contrasts. So in standard ASL methods, we acquire a labeled image and a control image and subtract one from the other to get our perfusion-weighted images. In contrast with VESPA, we need at least 4 images. So the velocity selective label and control is alternated, just like in normal ASL, while the PCASL label and control condition is alternated in a 2-label, 2-control pattern. Once we've done this, we can very easily separate our two contrasts in post-processing in a similar way to before with image subtractions. The advantage of doing this encoding and decoding step is that we get exactly the same information as if we did each labeling procedure separately, but we can do it in half the scan time without losing out on the number of averages we can acquire. So now we have separate velocity selective ASL and p-Casal contrasts decoded from a single scan. But we can do much better than just this qualitative information about CBF and transit times from this data. So here's a sort of a simplified version of the VESPA signal model where we're ignoring T1. And there's a graphical representation shown here on the right. So when the TI is greater than the transit time, we can calculate the transit time from essentially the ratio of the velocity selective signal to the p-Casal signal. And we can get the CBF from their magnitudes. We don't sort of do this sort of ratio thing in actual sort of, what we've actually done with the data is we fit it with a least squares fitting method. But that's essentially sort of how we get these two bits of information from our two contrasts. But something that's particularly cool with this method is when the transit time is longer than the TI, such as in cases of severely long transit times, we can still calculate CBF from the velocity selective ASL data. In this case, we can't calculate the exact value of the transit time like we could before, but we do know that it's longer than our TI. So by using the signal model, we can calculate CBF no matter what the transit time is, plus we get this information about transit time. Something that I also want to emphasize here is that we only need one time point to calculate both CBF and transit time using VESPA ASL. So this is different to spatially selective methods like p-Casal or PASL, where we need to acquire multiple time points to do this. So first, to evaluate this method, we did some simulation looking at different inflow times and their CBF and transit time errors. And the different inflow times are shown in different colors here. And these are sort of, the CBF errors on the left and the transit time errors on the right. So a TI of 1.4 seconds, which is shown here in red, has the most accurate CBF estimates in this case, but it can only estimate transit times up to 1.4 seconds, beyond which we only know that the transit time is longer than the TI. But for most experiments that we did, we decided to use a TI of 1.8 seconds, which is shown here in green, because it still has good CBF accuracy, but it can also estimate transit times up to 1.8 seconds, which should be appropriate for gray matter across most of the healthy brain. So we wanted to check this assumption and the results of our simulations in vivo. So for a single volunteer, here you can see quantified CBF maps on the left and transit time maps on the right, generated from about 3-minute VESPA scans at different transit times. So each transit time is shown on a different row and each column shows a different slice. You can see that the CBF maps are relatively consistent across transit TIs, only getting slightly noisier with a longer TI, as we expected. On the other hand, the transit time maps change quite a lot, up to a TI of about 1.8 seconds, after which they're relatively consistent. We can also fit this data together in a multi-TI fashion, which is shown here on the bottom row. And this confirms that most of the transit times for this subject are less than 1.8 seconds, apart from those in the white matter. So we continue to use TI of 1.8. So to validate the CBF and transit time measurements from VESPA to check, they were in the right ballpark, and they were accurate, we compared it to a conventional multi-PLD p-Casal protocol. So it has matched scan time and the scan parameters are shown here on the right. First of all, we ran some Monte Carlo simulations, and these quite nicely demonstrate the differences between the two techniques. So here are the CBF errors on the left and the transit time errors on the right. At very short transit times, the p-Casal CBF and transit time estimates are lower for p-Casal, so it's more accurate than the VESPA. But we can see at longer transit times, the p-Casal errors increase dramatically because the shorter post-labeling delay images no longer see any signal. In contrast, the VESPA errors remain pretty stable across transit times. And this makes it more robust to cases of unexpectedly long transit times. So next we ran the same comparison in vivo. So this is just showing a single healthy volunteer. And we expected both techniques to give similar information in this case, because with a healthy volunteer, they didn't have any particularly long transit times. So even though both techniques are very different in how they label the blood and how we calculate CBF and transit times from them, you can see that both give very similar information about these parameters. For example, both techniques show similarly long transit times in these border zone regions. So lastly, I just wanted to show you some of the other ways we can use the VESPA sequence. So at the top is the version we've already introduced, which can be used to generate robust quantitative CBF and transit time maps. A simple change to the sequence, which is shown on the second row here, is to turn off the vascular crushing module before the readout. This preserves the macrovascular signal, which is particularly evident in the p-Castle contrast. For TIs greater than 1 second, though, we can still get qualitative information about the CBF from the velocity selective contrast, because this doesn't have as much macrovascular signal in it. This might be a way to preserve the so-called arterial transit artifact, which can be particularly useful in diseases like arteriovenous malformations. Next is we have VESPA dual, which is these two last rows. So rather than varying the labeling control conditions differently for the two contrasts, for velocity selective and p-Castle, we can alternate them together. And this only gives us one contrast, but it has maximum SNR since the two contrasts are combined. We can either get maximum SNR perfusion weighted images by using the vascular crushing module, or if we turn off the vascular crushing, we can preserve the macrovascular signal. So just to conclude, VESPA ASL can give us quantitative CBF and transit time maps from a single scan. We validated the CBF and transit time maps from VESPA against a conventional multi-PLT p-Castle scan in a healthy volunteer. The advantage of VESPA ASL is that the CBF measurements should be insensitive to transit times, unlike with p-Castle or PASL. But we still get transit time information, unlike if we just performed a velocity selective ASL scan. And this makes VESPA a robust choice for quantifying CBF and transit times in a wide range of scenarios. In the future, we aim to validate the robustness of VESPA in clinical cases with delayed transit times. So with that, I'd like to thank my co-authors, the rest of the team at CFMRI, and thank you all for listening.